Hello? Am I broadcasting to the wide world of YouTube? Perhaps I am not. Interesting.
Hey guys, how's it going? Today I want to show you my awesome new Wix website. It's got everything I love all in one place.
I'm not. Oh, okay, it's going. All right, so I am going to.
Hey guys, how's it going? Today I want to show you my awesome new Wix website. It's got everything I love all in one place.
I think I have perhaps now sorted out the technical difficulties. It started broadcasting. I accidentally watched an ad on my own video, and then I watched myself watching the ad on my own video. So I'm just going to go ahead and hope that this is working now. And I see some hellos down in the comments. Yes, okay, so people can comment hello, so that I think means that this is working. So welcome to a super casual Saturday morning live stream. This is the first live stream that I have ever done. Oh, there are more of you. Hello, you're all saying hi. Oh my goodness, this is exciting. So what I'm gonna do today is I want this to be sort of like last year's 2017 community check-in. Which was a really nice way for me to sort of talk about where I was and hear from you guys about what you guys wanted, and then just also sort of talk about what I wanted to do in the year ahead. And so grad school has been crazy. And so rather than making a full edited video for that, I thought that a live stream might be a nice way to do that. So that's what I'm going to do today. We also have an activity. So somebody suggested, when I asked on Instagram for ideas of things that we could do today, somebody suggested that I should extract my own DNA. And I've done that in a video before. I put the link in the description box below, so that should hopefully be working. But that involves a lot of me spitting, which I think is gross to do live on camera. So instead, we are doing extractions from food. So strawberries are the typical thing that people generally extract DNA from. And the great thing about strawberries is that they have 8 copies of their genome. So we have 2 copies of our genome, but strawberries have 8, so they have way more DNA, so it's really easy to see when you extract it. But I asked what other fruits or foods I should try and extract from, and I got a ton of suggestions. You guys had lots and lots and lots of suggestions. So one thing that somebody suggested was bananas. And I happened to have a banana sitting in my kitchen, so we're doing a banana. And then somebody else, for a Valentine's theme, suggested strawberries and chocolate. Now, the thing about chocolate is that I had just grabbed a chocolate bar. There are a couple problems with trying to extract DNA from that. One is that the chocolate bar doesn't just have chocolate in it, right? So it doesn't just have cocoa. It also has milk, and it has sugar and lots of other things that will also have DNA. But the other thing about a chocolate bar is that it's been super processed. And so, you know, it's been heated, it's been broken down, all of this stuff has happened to it. So while there is likely some DNA in there, and I actually did find some DNA extraction from chocolate videos online, it's probably really degraded and really broken up and small. So I decided to try cocoa powder. I couldn't find any cocoa, any like raw cocoa beans, because that's kind of hard to do on one day's notice. But I am hoping that the cocoa powder is like just sort of not processed enough so that we can try and get something out of it. So it's going to be an experiment. We're going to see how it goes. But you guys have also sent in a bunch of questions that I have here that I'm going to try and answer as I'm doing DNA extractions. And so let's get started doing that. I also see that there's some comments coming in now. Ooh, okay. That's a technical DNA extraction question, so we'll get to that a little bit later once we start doing this. But the first thing I want to do is I want to just sort of update you on why there haven't been so many videos lately. And the reason for that is because grad school. So I have been trying to finish up my PhD. I'm really hoping to finish this summer, and it's been a lot of work. And so all of my time is going to lab right now. But somebody on one of these posts commented that they thought I had given up on YouTube, and that is absolutely not true. I have not given up on YouTube. And in fact, I'm still making YouTube videos. They're just not happening here. So I've had the really cool opportunity to work with a program called Genes in Space, which encourages middle and high school students to design genetics experiments that they then send up to the International Space Station. And I've been making videos with them. So I've made some videos about sort of typical lab experiments. So things like PCR, things like gel electrophoresis. And I'm also going around and interviewing scientists for them and with them. And it's been so much fun. And so I've actually been putting videos out on YouTube, just not here. So all of my current extra video time has been going to that project rather than my own videos. So you can check those out. And I hope that you would. I've been putting a lot of sort of time and love into those, and they've been really fun. And they cover some of my favorite topics like science and scientists and DNA. So I put a link down to the Genes in Space YouTube channel below, and you can go check those out if you're interested and if you want to see more videos of me. But hopefully soon I'm still working on that project, but it's going to wrap up kind of soon. Oh yes, and somebody just commented, please don't stop making videos after your PhD. No, the idea is that after my PhD, I will start making lots of videos. So I just need to get this sort of portion of my education finished. And then be able to move on to making lots and lots and lots of videos. So many videos that you're going to feel inundated with videos, because I have all of these ideas that I've been trying to do for the past year or so and just haven't had time to do right and to do justice to. Because when I make a video, I really want it to be high quality. I want it to be good. And so I want it to be something that I'm proud of and that you guys are proud of sort of being a supporter of. And so there are more videos coming to this channel and to other places soon. I just need to do a lot of science first. Okay, so I want to jump into some of the questions that people have sent, but I also want to get the DNA extractions going, because this is actually something that I do a lot in the lab, is I extract DNA out of stuff. But I work on human genetics. So I'm usually extracting human DNA, but today we're going to be doing some foods. And so I want to get this sort of started as we're going. So the first thing that we are going to do to our strawberries and then to our other foods is we're going to add our lysis solution. Now what this is some warm water that I have mixed with dish soap and salt. So what this is going to do is the dish soap is actually going to disrupt the cell membranes of the fruit, and it's going to help break open those cells and help them to be able to actually release their DNA out into the solution. And the salt is going to help any sort of proteins that are stuck onto that DNA release, so that we can actually sort of get those down into the sort of waste portion of the solution and then just have the DNA. Now, the one thing I want to try and do, and you're going to have to bear with me, we're going to see how this works, is I want to actually zoom you guys out so that you can see a bit more of what I'm doing here on my desk. So I'm doing this off of my laptop right now, but I have these little iPhone lenses, and I have a wide angle lens here, and I think this is gonna work. I tried this out earlier. If I put, oh, that looks very blurry, but I think we can get this kind of set up here so that, all right, we're a little more pixelated, but now you guys can see my desk. Let me know if you think this is terrible, but this way you guys can see my desk, and now we'll be able to sort of look at what I'm doing. So I have my strawberries here, and I'm going to add my lysis solution. Okay, not too much. And now I'm going to take a muddler, and I'm going to start muddling my strawberries as I answer the first question that I got. And I actually wasn't sure if people wanted me to use their names attached to these questions, so I am not using your names, just in case you wanted these questions to be private. So the first question that somebody asked is, what music do I jam to while I'm working? So this very much depends on what I am doing in the lab. So sometimes I am in the lab and I'm doing something where I have to count a lot and I really have to pay attention to what I'm doing. And so in those moments, I often listen to white noise. So I think I talked about this in a video at one point where I listen to like Spotify focus playlists. And so they've just got like ambient music, and it's, you know, sort of dreamy ambient music. I don't know, it's very indie hipsterish. And I have no idea what any of those songs actually are, but that's sort of good if I'm like loading a qPCR plate and I have to be counting things and I don't want to be sort of distracted by words of a podcast, because the rest of the time when I'm in lab, if I'm doing really repetitive tasks that I don't have to, oh, hello, more people are joining, hello. If I'm doing stuff that I don't have to really pay attention to, I'm often listening to podcasts. So if I have to just, a lot of my job is moving liquids back and forth. over and over all day. And so that gets very repetitive. And so I listen to a lot of podcasts to keep me sane and make me feel like something stimulating is happening to my brain. But then if I am doing something where I am trying to really focus on writing, then I listen to actual white noise. And so I listen to something called Noisely. It's NOIS, I think .li. I think that's how it is. I just Google it every time to get there. And so Noisely is really great for total background noise to block out anybody in the lab who's sort of talking or chit-chatting or any sort of noise from the refrigerator and all that kind of stuff. So that's really great for just sort of blocking out noise. Okay, I have like macerated these poor strawberries a bit. So I'm actually going to let them sit in the solution a little bit to let that lysis buffer work. And I'm going to clean off my muddler before I go, ooh, I'm making a complete mess. I'm going to clean off my muddler before I go to the bananas. And actually, so somebody just asked what podcasts, and then somebody else responded thanking me for introducing them to Sawbones. And in fact, that is one of the podcasts I was going to tell you about. I am obsessed with Sawbones. It is a podcast by a husband and wife. The wife is a doctor, and the husband is a comedian. He's like a professional podcaster. It's Justin McElroy. And so I love it because they do tours of like medical history, and it's so much fun because, you know, it's funny, and they talk about how we've treated different diseases through the years, and they talk about sort of medical mysteries, but now they've also, I think they're running out of some medical like history bits a little bit, but I still love it. But now they'll do things like they did a recent one on antibiotics and sort of the history of antibiotics. They've done some so topical things. They actually just did one on Tide Pods, I think. So, you know, topical, fun history, science, medicine stuff too. So that's just a lot of fun to listen to. And it's sort of friendly too if I'm just bopping about in lab and I can sort of listen to people talking about funny medical history science. Okay, the bananas were much easier to mush up than anything else. So Caden Erickson just said, it's okay to extract DNA at home as long as you clean up and are careful with disposal. That's what my bio teacher said. And yes, so actually one of the reasons why I'm doing it this way is because all of these things are things that you have safe at home. So, you know, you're extracting DNA, but you're eating DNA in these foods all the time. DNA is not harmful. And none of the things I'm using are harmful, right? I've got salt water, I've got soap, I've got, well, okay, now in these days, I can't say that soap isn't harmful because of the crazy Tide Pod stuff, but you know, as long as you're careful, right? Like, don't eat any of this. Don't eat any of this. Okay, so, and now I'm going to do the cocoa. And the next question on my list here that somebody sent in is instances of encouragement or discouragement and how I have reacted to them. So somebody asked a question about if there were any really big times where somebody either said something encouraging to me. Oh, okay, the cocoa powder one smells amazing, but looks kind of crazy. If somebody said something encouraging or discouraging to me and what that, you know, what effect that had on me over time. So I actually made a video about this, my I'm Here to Learn the Hard Way video, where I talked about a time where I was in lab and somebody said, oh, have you tried, you know, this protocol? And I said, oh, I haven't heard of this protocol before. And their response was, you know, a second-year genetics PhD student at Stanford should know what X is. And I didn't. And that was really, really discouraging to me because it just made me feel like maybe I didn't belong here, you know, and I just felt so bad about that. But my response to that was, again, to remind myself that I am here to learn. And if I knew all of these things, then you know, I wouldn't need to be here. I would already have my PhD. So in all of those discouraging moments, and there happened to be a lot of them in grad school, there are a lot of discouraging moments. It's, you know, it's nice to just sort of remember that why I'm doing it. So I think that's my tip for if somebody says something very discouraging to you. I guess I just remember why I'm doing that thing in the first place. I'm likely not doing it to impress that person, right? Like nothing that I'm doing, especially in grad school, is to impress anybody. It's for me to learn. That's why I came here. I don't want to be a career researcher. I want to go into science communication. And so, hello, I see more people, hello. And so I am here to really learn and to absorb as much information as possible. And so, you know, those times where somebody has said something, I try and look at it as a learning experience. So that's one thing. But moments of encouragement. I mean, I think I'm one of those people who I don't always listen to the moments of encouragement. I can be very self-deprecating, but I'm trying to be much better about if somebody says I've done a good job at something or somebody's trying to be, you know, really encouraging. I'm trying to do a much better job of taking that in and really accepting that. So yeah, figure out what you're really doing, what reason you're really doing the thing that somebody said something discouraging for, and then try and be better about accepting those sometimes rare moments of encouragement. Okay. Oh, somebody's asking where to send your questions. I'm trying to watch the comments as I'm doing this and talking. So you can send your questions in right here, and I will try and add them to my mental list, and I will try and get through them. Oh, what is the ratio of salt water and soap for that solution? So what I did is I had 2 cups of warm water. I added 8 grams of salt, and then I eyeballed about 15 milliliters of, 8 grams of salt, and I eyeballed about 15 milliliters of soap. I sort of poured it until it looked right. So your results may vary. Okay. So next step, before I answer more questions, I'm going to try and filter out all of the extra gunk in here. So I just have a coffee filter over a water glass, and we are going to hope that the coffee filters don't break like they did when I did a test one. Okay, so I'm just going to let this strain off to the side for a moment. Okay, so, somebody just sent in, I'm about to be a senior in the anthropology department, and I often feel like I'm not inquisitive or passionate enough. That is one of, I think, my biggest things that I think about myself all the time, is that I'm not creative enough or inquisitive enough for science. And I think that that's part of the nature of being in programs like this, is that, you know, you see all these super smart people around you, and, you know, they come up with all these great questions. And I really think that asking good questions is the key to life, not just to science. But I think that, you know, I'm going to water down the chocolate a little bit to see if that helps it run through the filter. I think that just remembering that, you know, you had some sort of question and some sort of passion that brought you here, that brought you into this field. And so, you know, you're here for a reason. You decided to come into this field for a reason, and you're always going to see those people around you. But this is part of imposter syndrome. Those people are probably thinking the same things too. And so trust the ideas and the passion that you have and the questions that you're asking, and just sort of go for it. I mean, because you can self-doubt yourself. I do this all the time. You can self-doubt yourself forever. But I think just sort of remembering the passion that you have that brought you to this point can be, for me, important in tackling those. That, you know, I didn't just wander over here, right? Like, I had questions and I had passion, and that's why I'm here. And so try and trust in that. Okay, so Pharmacon has asked a couple times, so I'll get to it. I have not already answered your question on PhD versus MD. So I am in a pretty translational lab. So they asked, you know, if they're interested in translational science, is it better to do sort of PhD or MD? And I'm in a very translational lab. So we work on human genetics, and I'm thinking about human biology all the time and trying to think about ways that we can use our research to sort of better human health. And I would say, oh, okay, I'm spilling chocolate soap water everywhere. That's exciting. So I would say that both can be really valuable, and it's just what you want to do with it, but you also don't have to choose. So my advisor is an MD PhD, and I know a lot of MD PhDs at Stanford. And so if you really want to straddle that line, you can do both. But if you do only want to do one, because MD PhD is a lot of years of school, I mean, I think thinking about what parts of it you love is really important. So do you love sort of the, you know, interacting with patients, that sort of one-on-one interaction of really helping people sort of in that day-to-day? Do you love that really hands-on aspect of it? Or do you want to be like me right now, sort of more behind the scenes in the lab, sort of asking the much more basic questions? Because I think that the nice thing about sort of being in a translational lab is that I do sort of get to see, you know, a bit of this patient interaction by watching the doctors in my lab, but I'm not sure that I would be the greatest doctor. But they get the fun of, you know, when something actually works and it goes through clinical trials and it goes through this whole long process, they get to then actually bring it back to the patients and sort of bring all these revolutionary things to them. Whereas the stuff that I am working on, if it ever makes it to a patient, is like 10, 15 years down the road, right? So I'll never get that moment. but I sort of like the tinkering little bits of it. So this is a very rambling answer. Clearly my like live stream skills need to be reined in a bit, but I think you don't have to choose between MD or PhD, but think about the parts of it that you want to do on a daily basis. Do you want to be interacting with patients every day as an MD, or do you want to be working sort of behind the scenes in the lab every day? Okay. And people are asking about why there's soap in the water. So this is because, yes, so somebody just said it's the phospholipid bilayer. So the cells are surrounded by this lipid bilayer, and plant cells also have a cell wall. And so the soap is helping to break through that and break that apart and really help to release the DNA out into the solution. Oh, and I just realized you can see my like giant collection of glassware here. I was worried that these might not work, so I pulled like all of the glasses in my apartment out. I wasn't drinking 5 glasses of water. Okay, so the next, have I seen House MD? I have seen House. I used to watch that in high school, I think, maybe. Yeah, I have seen House. I'm definitely no House. I am not that smart. Okay, so what's another question? So somebody asked who my favorite scientist is, and I have so much trouble answering this question because I have like 400 favorite scientists. Every time a new scientist comes to speak at Stanford, I like decide that they're my new favorite scientists and they're so cool. So I don't know that I have a favorite scientist. I did talk about in a video a while back that I made for some collaboration with, I think It's Okay to Be Smart, like Elizabeth Blackburn is one of my favorite scientists, and she is because she sort of discovered and did a lot of the basic work on telomeres and sort of looking at these little end caps on your DNA that help protect them as your cells are replicating. But and that was, that was like 4 or 5 years, maybe 4 years ago. And I was like, yes, she's my favorite. But then probably the next week I was like, no, there's another favorite. And so I don't know that I have a favorite scientist because I bounce around both between like the big names that sort of come and talk at Stanford and also between, the amazing people I meet every day. I'm just like always in awe of, the postdocs and the grad students that I work with. And I don't know, that's a lame answer to who's my favorite scientist, but basically everybody, which is why I want part of my future career to be interviewing lots and lots of scientists. Okay, you're all commenting, yes, I know this is a wine glass, but I do also drink water out of it sometimes. So that's part of the reason why I want to be interviewing scientists as my career, because I just love talking to scientists and I think that they are so smart and so passionate about what they do. And I want to talk to all of these smart, passionate people and hear them tell me about their cool, awesome ideas basically every day of my life. So I have many favorite scientists and I hope to meet many, many more. Okay, so our kind of gross looking solutions here have pretty much stopped straining. What I'm going to try and carefully do is I'm going to take the filters full of the like fruit schmush, and I'm going to try and move them into these little buckets without breaking the filters, like I did when I did a test one about an hour ago. So that's fun. Oh no. Okay, we're gonna tip this on its side and pull it this way. Okay, oh my gosh, so much mess. Okay, one thing I want to mention about this too is that I'm doing this with all things that you likely have at home, right? You might have soap and salt and all these kinds of things. This is really close to what we do in lab. Now we do it with really expensive kits because they all have proprietary reagents, and they're very fancy salts and very fancy soap. But it's the same process, right? We take some buffer that has like salts and some soap in it. We break open the cells and then we basically we precipitate and then wash the DNA. So that's the next thing I'm going to do is precipitation. But I want to get all of these little baggies of, oh no, don't fall in, little baggies of fruit out. Clearly, super professional live stream activity happening here as I baggie of fruit. And oh, the chocolate one looks terrible. I don't know that this is going to have worked. I did find some videos online from like chemical supply companies about how to do chocolate extraction from samples like this, but they did require all of those really fancy kits. And so that's the thing, is that the ones we do in lab, it's really the same principles, but it's a lot cleaner. You have a lot more control than I do here with my soap water and my salt. But again, same concept. Okay. Oh, your 5-year-old daughter is so interested in this right now. Yes. Hello. Oh my gosh. So that's the reason why I want to do it this way too, is because you can do this at home. You probably have these things. So what I'm doing back here off screen is I have a bowl of ice water that has isopropyl alcohol in it. So the alcohol, it's cold, which is because it was in ice water. DNA is soluble in water. So this is mostly like a water solution right now, or it's an aqueous solution. But DNA is not soluble in isopropyl alcohol, or it'll precipitate out. So what I can do is I'm going to pour in the isopropyl alcohol, and there's going to be a layer in between the alcohol and the like fruit smush layer. And so the DNA that's right on there will precipitate out at the edge. And again, this is what we do in lab. We take either isopropyl alcohol or sometimes ethanol, depending on exactly what nucleotides, DNA or RNA we're going to look at. And we pull out the DNA this way. In fact, right now, sitting in my freezer at lab, I have some RNA precipitating in some ethanol, so not isopropyl alcohol, but a different type of alcohol, and waiting for me to go in and clean it and wash it up later today. So again, this is just what we do. And also for that sample, I was not using a muddler, but I was using, to break up the sample, a tall tube and a sort of little homogenizer that went in and out of the tube, so like a mortar and pestle. So again, very similar to what we're doing. Okay, so what I am going to do is I'm going to very carefully try and float. See, I'm making a mess already. I'm going to pour this isopropyl alcohol down the side and try and kind of float it on top. Okay, and oh my gosh, there's so much. Can you see that there's that like bubbly layer in between? That bubbly layer is the DNA precipitating out, which is crazy. I'm spilling isopropyl alcohol on my laptop. Let's clean that up. So you can see that layer. And I'm actually wondering, see, we have these fun little iPhone lenses here. If I take off my wide angle, it might be a little easier for you to see. Let me switch the camera view so I can see. Oh yeah, so can you see that like mass that's floating up there now? That is the DNA. So, oh my gosh, it's very sticky looking. So I'm gonna take a chopstick, or not a chopstick, I couldn't find chopsticks. It's a skewer, and I'm going to pull up this like mass of DNA. That is crazy. So strawberries have tons of DNA. This is why this is how this demonstration is often done, is with strawberries, is because there's so much DNA. So this is a chunk of DNA. So here's what you can give to your Valentine on Wednesday, is a sticky clump of strawberry DNA and maybe some chocolate DNA if we can get that out. So that's a thing. Okay, so cool. So somebody just asked, is that just DNA or is it all the protein too? It's likely some protein as well. So certainly the salts, we're trying to get some of the proteins to stick or to unstick from the DNA. But again, this is not the cleanest preparation. There's probably some other stuff stuck in there too, but mostly DNA. So this is crazy. So now I want to see if it works with the banana too. Okay, I'm going to leave you sort of zoomed in for the moment. So, okay. I'm going to pour on the alcohol. And I think that's forming. It's a little harder to see with the yellow. Okay, let me get a skewer. Okay, and oh, okay, so this is not as sticky. So likely there was a lot of sort of protein and sugars still stuck in that DNA before, because it sort of came up as a glob. But can you kind of see? Oh, yeah. So you can kind of see there's like some stringiness here happening, right? So there we have some strands of DNA, which is pretty crazy. This is banana DNA. So yeah, so it's not the cleanest DNA preparation. Oh gosh, okay. It's not the cleanest DNA preparation that has ever happened before. There's definitely going to be some other stuff stuck in here, but you know, there's a lot of DNA in there too. Oh, so the yellow, this was a banana that I strained. All right, so now let's try the chocolate. I do not think that this is gonna work, but we're gonna try it anyways. And yeah. So chocolate might require a little more careful preparation. So we are seeing a little bit of a separation of the isopropyl alcohol layer. But I cannot confidently say that any of the stuff that we're seeing in here is DNA. It might be, but I'm not going to confidently say that we have extracted beautiful chocolate. Yeah, there's not much happening here. So, and the reason for that too is that, you know, DNA is in every living thing that we know of here on Earth. And so DNA is in your food, but when you get to the really processed foods, it's likely been so broken down by like salt concentrations and heat concentrations that it's not really tons of DNA. If there is DNA left in there, it's probably really broken down into very, very small pieces. And people are asking what it tastes like. I'm not going to eat it because it's got a ton of soap in there. So, you know, processed foods likely still have some DNA in it, but it's going to be really broken down and degraded, whereas fresh foods, and especially things like strawberries that have lots and lots of copies of their genome, are going to have way more DNA that's still in sort of big, long strands in there that you can actually extract and see. So yeah, so it's fun. So, and again, you can do this with your own cheek cells. So in the video down below, what I did is I took just salt water, just salt and water, and I swished it around and spit that out. And then I added soap to that, and then, you know, I mixed and then I added in the isopropyl alcohol, and you can get out strands of your own DNA too. And again, that's also not super clean, right? You're also probably getting some DNA from the bacteria in your mouth, so it's not all just from you. You know, there's DNA in your food, and in stuff like strawberries, there's so much of it. And actually the banana too. I'm not sure if there's a lot in the banana, and I'm not sure if that's because it's really a super unclean prep, or if there's also, or because I just used a lot of banana because I was not expecting that much. So yeah, so this is our DNA extraction, and I'll do a couple more questions. Excuse me. Oh, how much student loan debt do I have? I am thankful that I am in a position where I don't have any student loan debt. So I ended up going to undergrad with a big scholarship, which was really, really helpful to me. And I'm very glad that I chose the school that gave me a lot of money, both because it wound up being a great school, and also because I now am not, as many of my peers are, really struggling with student loan debt. So I am so thankful that I both had a big scholarship and that my parents were willing to pay for my room and board while I was in school, and were very helpful in doing that. And then thankfully for grad school, for my PhD, this is also funded, and so I'm not going into more debt here. So I so appreciate that I am in that position, because many of my peers are not, and are really struggling with student loan debt. So that was sort of a random out of the blue question that just popped up. Okay, so I'm a third-year student in maths and computer science. I don't know if I want to get my master's, PhD, or get a job. What are your plans when you finish school? How do you plan? Okay, so actually this ties into a question that I got before as well, which was when was the moment that I knew I wanted to go to grad school? And I actually don't know when the moment that I knew I wanted to go to grad school was. I think that as growing up, a lot of the people that I knew who had finished college had also gone on to get some sort of advanced degree. So either a master's or a law degree, or had become a veterinarian. So like a lot of the members of my family and a lot of people that I knew also had additional degrees. So it was sort of a not expected thing, but I think it was sort of normal that I had considered that grad school might be an option for me. But the moment that I do remember is when I realized in college that it wasn't the right time for me. So I took 2 years off in between undergrad and grad school, and many of my friends just went straight through. And at the time where they were all starting to apply to graduate school, I just sort of knew that I wasn't ready. And I worked really, really hard in college, and I was sort of getting to a point where I was worried about sort of burning out a little bit. And I loved science and I loved the work I was doing in the lab, but I just kind of wanted a couple years to do something different and also to get hands-on film experience, because I had a lot of really great hands-on biology experience in undergrad. But even though I had a film degree, I only had a little bit of hands-on film experience. So I wanted to go out and get some more work done with that too. So I ended up taking a year off before I applied to grad school. And I think that was really important for me. And it's not for everybody, but having that year to sort of have a real job, go out in the real world, have a 9 to 5 and work and sort of manage myself and my life and just sort of be an adult for a couple of years was really good. And in fact, I got into grad school and then postponed and deferred my admission for an additional year because I decided that I had so much more to learn about the job that I was in, and I really had a lot more to absorb from that. And so thankfully my job let me stay for another year and my grad school let me defer for a year. And I so appreciate both of those places for doing that for me, because I think that those 2 years were really important to sort of my like adult maturity development. I think that was really important to me to just sort of get out of science for a little while and gain some perspective and then come back in. And when I came back in, I was really sort of excited and refreshed and recharged. Whereas I feel like if I had gone straight from undergrad, I did feel kind of burned out at the end of undergrad. And I think I wouldn't have been as successful, not that I've been super successful in grad school, but I don't think I would have been as sort of maturity-wise successful in handling sort of the ups and downs of grad school if I had gone straight from undergrad. So those 2 years were really, excuse me, really, really important for me and sort of my career development. And so somebody also just asked a question as a follow-on, can you describe the learning curve from undergrad to grad as in difficulty of classes and time management? And I think that for me, the biggest difference between there are a bunch of big differences between undergrad and grad school. And a lot of the questions I've been getting have been about grad school. And I think the big one is that for undergrad, you sort of have set courses and you take those courses, and depending on what sort of system your undergrad is in, you either take them for a quarter or a semester. Maybe you have a year-long course that follows on, and then you're done, and then you move on to the next thing. And it's these really defined chunks. Whereas in grad school, you take on a project and then you work on that project for 3, 4, 5 years. And so there's no defined endpoint and there aren't defined goals. There's not a midterm. There's not sort of a paper due. So you really have to set those goals for yourself and say when you're going to get certain things done. You really have to be good about time management because it varies lab to lab as to how involved your advisor is going to be in your sort of day-to-day time management. But most labs, you set your schedule. And so there's nobody really checking up on you. There's nobody who's going to come in and say, you know, did you do these 3 things? Are you on track? Are you on time? So you really have to be great about time management and also great about setting goals for yourself. That's been one of the biggest things for me, is setting concrete milestones for myself and then trying to achieve those milestones because you don't have them. And the other big thing, I think the thing that really sort of hit me in the face when I showed up to grad school is that for undergrad, when you need answers for a class, there's a textbook or there are required readings or there's a defined place where you go to find those things. And in grad school, there's not. And so you come across a problem and you have to search the world to try and find a protocol that will help or a paper that will help or something that will sort of really give you a better sense of what you're doing and sort of what you need to get done to move forwards in your project. And so that to me was sort of a huge revelation that, oh, like when I was taking classes my first couple years of grad school, there wasn't a textbook. When I wanted to figure out the answer for something, I had to go search for whatever the right paper to do that was. So that to me has been sort of a big, or it was maybe my first like 6 months was sort of a big learning curve of how do I figure out where to find the answers and what do I do? But then I figured it out and I learned. And so that got a lot easier. Okay. So have you written articles? So I wrote a review article that should be hopefully coming out soon. And I'm trying to make a video on. I have like 3.5 videos that are filmed or some way filmed that just I have not had time to finish. But I wrote a review article that should be coming out soon. And then I'm working on 2 scientific sort of manuscripts right now that will hopefully be published at some point in the next year or so. I want to get both of those done before I graduate in the summer. I'm hopefully graduating in the summer. So yeah, so no published articles yet, one that has been accepted as a review, and then 2 that are hopefully coming soon. Okay. Somebody asked before the live stream what my favorite video that I've made was. And this is hard to choose. It's like choosing your favorite children. And so one of my favorites for a long time was my film noir alleles video talking about sort of blue or brown eyes. And that one was so much fun just because like it was great to do something really different in that video. I like made myself up as 2 different characters, and I wrote out a very dramatic script, and I tried to do film noir lighting, and all I wound up doing was almost setting one of my light boxes on fire. So the lighting wasn't what I had hoped it would be. But it was just sort of fun to play with sort of set pieces and really more dramatic things. And I'd love to have time to do more of those, hopefully again after grad school. But now, one of my most favorite ones is the interview I did with astronaut Kate Rubins, because I got to interview an astronaut, and I would never have imagined that that was possible. And oh, it was so much fun I was also so amazed because I reached out to NASA and said, hey, can I interview her via Skype? And they were gracious enough to say yes to that. And that was amazing. I did not think that they were going to say yes to some random person asking, hey, can I interview an astronaut? And that was a huge compliment that they also thought that I was capable of doing that. But then I went farther and I asked, hey, can I come to Houston and interview her in person, and maybe we could sequence some DNA together? And they said yes to that too. And that to me is such a huge, amazing thing about NASA, because I expressed my sort of delight over this and my surprise that they would take the time to actually, you know, have multiple scientists sit down with me and talk about the projects they were doing. People's time is so valuable. And so the fact that they actually you know, all of these scientists and astronauts were so giving of their time to me to sit down and talk to me was huge. And everybody made the point to me that that's part of their mission, that NASA, you know, at least for us in the US, NASA is our sort of space, you know, administration, society, space organization. And that part of their goal is to communicate what they're doing to the public. And so that was just so, I don't know, that was moving to me that they took the time to talk to me, and we're all so nice and so amazing. And so I really, really appreciate that that happened. So that's also one of my new favorite ones. Oh yeah, never be afraid to ask. Yeah, absolutely. That's another thing I've learned over the past year, is that with, I think, one exception, every time that I have reached out to a scientist and said, hey, I want to make a video about the cool thing that you're doing, would you talk with me about it? I think once somebody has said no. A couple times it hasn't worked out because of timing, but people say yes. And that's, again, so huge to me that people are willing to take their time to talk with me and, you know, let me make these little videos about the science that they're doing. Also with the DNA data storage, that was completely a cold, you know, reach out question that I just reached out to these scientists and said, hey, you published this cool paper. Can I Skype interview you about it? And Dina said yes. And again, I was so just honored that she gave her time to let me babble about her science with her. So that was really fun. I know I'm missing so many questions. There are a lot of questions. A lot came in beforehand and a lot are coming in now. So I'll maybe stick around here for another 15 minutes or so until about 11 and answer a couple more. But I do want to try and get to some of the ones that are coming in now and some of the ones that came in before. So somebody asked a question. Have you ever thought about making a motivational video for teens or children, like why is it cool becoming a scientist, or how did I end up being a scientist? And I've actually thought a lot about doing another one of these, because I made one before I came to grad school of the sort of my path to where I was at that point. But it's changed a bunch now, right? So now I'm in grad school and I'm looking forwards to a career in science communication. And so a lot has changed. And so I do really want to make an updated video of sort of how I got to here, because I love hearing those stories of how I got to here. And actually, I don't know if she's still here, but Sally LePage was here for a bit. And I actually sat down for a sofa science video with her. And so that's actually a more recent updated version of how I got to here, was the video that she filmed with me as we were sitting on a sofa and talking about science in our lives and stuff. So that's actually a more updated one, but I should probably do one on my channel too. But the other part of that question was, why is it cool becoming a scientist? And I think for me, that's because I get to ask questions all day. And sometimes that's discouraging when I don't get the answers to those questions or my experiments don't go well. And that's another question that I'm going to answer quickly. But I just get to ask questions all day and really sort of play in the lab with trying to figure out the answers to those questions. And so that's so much fun to me. And that's one of the things that I try and remember. Somebody asked, how do you deal with failed experiments? One of the things that I try and remember is that it's fun to ask the question. So I am currently in the middle of a lot of failed experiments, and things don't always work right on the first try, and things don't always go well. And I think there are sort of two things depending on the day. And one of those things is sometimes the correct answer is that it's 11 o'clock at night and everything has failed. And if I do more, I'm just going to mess it up. So I just need to go home. I need to sleep. I need to, wake up the next morning and have a nice fresh cup of coffee and recenter and then go back and tackle it in the lab. So sometimes that's the answer for me, is to just step away from it and think about it. Because I find, and I actually talked about this in a video too, my sort of doing versus thinking video, that if I've had, a big string of failures, I sometimes find myself in this repetitive circle where I just keep trying and trying and trying and trying without really stopping and thinking about what I'm trying and if it's going to solve the problem that I'm having. So sometimes the answer for me is sort of to stop and step back and move away. The other option sometimes is that I do need to just keep trying the next thing. So I've had a series of small failures in the lab this week where I'm, in fact, instead of a DNA extraction, I'm trying to do an RNA extraction from a very difficult sample, and it's not going well. And so I am in the mood where I'm throwing myself into it. And so I am trying lots of different measures, and I'm trying to get it out, and I'm doing my best, and I'm just tackling with different strategies. But I'm also asking questions, so, of other members of my lab. I've been maybe perhaps too vocal about my failures on this specific experiment this week, but I've gotten lots of great suggestions from my lab members. And so it's good to sort of check in with people and be like, okay, this failed. And I did it this way. What way would you do it? And how have you tried it? And so that's another thing, is if you're going to sort of go into that do mode, it's nice to also sometimes just bounce ideas off of somebody else of, here are all the different things that I'm trying. And so what do you think? And that can be really useful to trying to get over those failed experiments. But I take it personally sometimes if an experiment fails. I will feel bad for days that, maybe I did something wrong, or I don't know. I do take it very personally, and I'm trying to get better about not taking it personally and being able to separate sort of my emotions from the success or failure of my experiments. But it's hard because I'm tackling these questions. I've been working on this project for years and I want it to work. And so it's very hard sometimes to try and separate those two things, but I'm trying to be better. Okay. So, oh yes, people are asking if I will upload this afterwards. I will. I think that it automatically just goes live. And so I'm going to let it sit on my channel because I haven't done anything too embarrassing today, so why not let it live forever on the internet? Okay, so somebody just asked about the social life at Stanford, and that people don't even take the time to eat lunch together and those sorts of things. I think that it's hard for me to comment on that because I have such a singular experience of sort of my lab and my department in the School of Medicine as a graduate student. So I cannot speak for all of Stanford, but I actually think that my department does a pretty good job of sort of keeping in touch with our classmates. And, it's hard because people get busy, but I see sort of my year level classmates a bunch and, sort of maybe once a month, and I see my lab mates much more regularly than that. And, some of my friends will go hiking. And so we do sort of have, people are busy, but I think that for me in my lab and my department, we do a pretty good job of trying to also keep social connections and social lives, because that's an important part of grad school, I think, is having a good support network. And part of that is, outside of Stanford. So sort of, my friends and my family and my significant other who are outside of Stanford, but then also a good sort of support network inside Stanford. I think both of those things are important, at least for me, to my grad school success. Somebody just said, okay, here are two questions that I'm going to answer. So somebody just said, they enjoy my travel vlogs, and you're in luck because I am so excited. I'm going to a conference next week, and I'm going to make another conference vlog. Again, don't know when I'm gonna edit it because I filmed a week in the life vlog 3 months ago now that I still haven't edited, but I'm gonna try and do the travel vlog really and be on top of it because I'm going to AAAS in Austin. And I am so excited because it's sort of a combination of science and science communication and science policy. And I am so, so, so excited to go to this conference. I've never been so excited to go to a conference before. So I'm going to try and vlog as much of that as possible. So there's hopefully another travel vlog coming your way soon. Okay, and then somebody asked if there's going to be a follow-up to the Aaron Carter montage. And I have now gotten this question, I think, 4 times for this live stream. Most of them came in beforehand. And it's a very fair question. So I did film year 26, but I was in a rough grad school patch at that point. And so I was very grumpy, and it shows in that 26-year video. And so I'm gonna keep filming them. But I think that, you know, maybe that'll come out with 29, or maybe, you know, that'll come out with 32 or something. So, you know, it's been filmed. It will happen at some point. We just need to get, you know, a high enough density of happy ones in there. But I turn 29. Oh my gosh, do I turn 29 in like 2 months? In March? Yeah, I turned 29 this year. I've actually hit that point in my life where I'm like, how old am I? Yeah, so I turned 29. So the 29 one could be filmed coming soon, but 26 is not going out by itself because I was real grumpy. And I think that that's, you know, that's fair. It shows many stages of my life, and that was the grumpy stage. Okay, so somebody else just asked, how did you come up with the idea of starting your own YouTube channel about science? Oh, I'm so excited that inspired you to start your own in the future. So again, as I mentioned actually earlier in this, I took a couple years off after graduate school, after undergrad, before graduate school, to work in a production company for a couple years. And I loved that, but I missed talking to people about science. And so it was sort of a outlet for me to talk to people about science. And actually, I mean, the true story here, right, because we just talked about the Aaron Carter montage, is that I made that and I sent it out to a couple people thinking that like my friends would find it funny. And then that went big and a lot of people saw that. And I'd had this idea to start a science education YouTube channel. And suddenly I had a YouTube channel with a bunch of subscribers, and all of those subscribers showed up for an Aaron Carter montage, but I had a captive audience and I was going to teach them about science. And so this is one of those like unreleased videos. The first video I put out was the brain freeze video. That was the second time I made that video. I had made it earlier, you know, maybe a year earlier actually, because I filmed it in the house that I lived in my senior year of college. And I'd been playing with this idea of starting a YouTube channel for that long. And I'd sort of decided to change the style a little bit when I re-recorded it for the one that actually went out on my video. But it's the exact same script, and it's almost the exact same timing. So I should at some point put together the 2 videos side by side, because I might have to make 1 or 2 edits, but they're very close, but also very different styles. So that could be kind of interesting to look at. So that was a very long answer. What specific goals do I have as a science communicator? So as somebody who I'm now hopefully soon getting my PhD in genetics. And so one of the things that I really want to do as a science communicator is sort of educate people about genetics, because I think that more and more people are going to be facing decisions about genetics in their everyday life. So medical genetic testing is becoming more and more common. So people are going to start being asked by their doctors if they want to get genetic tests. People are facing things like GMOs in the grocery store and are getting sort of mixed messages about what genetic modification is, or is it not safe, right? There's a lot of conflicting messages out there in the world right now. And also, you know, things like CRISPR are coming up and popping up, and we as a society are going to have to start, you know, asking questions about how we want to use that and how we want to use it in medicine, or all these kinds of things. And so I think people are going to start increasingly facing genetics more and more in their daily life. But I don't think that, at least for me, I got very sort of basic genetics in high school, and then I took specific genetics courses in college, but it's, you know, it's not something that I think, if I had decided to go into literature instead, right, the only exposure I would have to genetics is that little bit that I got in high school. And I think that people deserve more, and they deserve more fun ways of getting access to genetic information, right? Like, it doesn't have to be this scary thing, but I think that any new field sounds really scary when you first hear about it. Like, people, whenever I hear something about physics, even in the excuse me, even in the news, I get a little scared because that's not my field, and I don't know if I'm going to understand everything that it says. So I want to make sort of really fun ways for people to sort of be introduced to genetics, and then also some great resources so that when they have questions, they can come and find answers to them. So yeah, so that was a very long babble about my specific goals are to really make genetics more approachable and accessible, and to make some great genetics resources so that when people have questions, because I think they're going to start having these questions more and more, they have sort of a safe, reliable place to find those answers. I hope that was enough of a babble about that. Okay. Ooh, are there any of your videos that you'd like to redo, either because it's too cringy or the science talked about it or the science talked about in it has changed? And actually, yeah. So I'm not going to point out which ones are the most cringy, because then you're going to go back and watch them and see how cringy they are. But a lot of the early bite-sized videos I was still working on sort of what my style was and sort of how, ooh, my window just made a noise and it should not have made a noise. That's weird. So I was still working on the style, and I loved the pink glasses at the time, but looking back, I cringed just a little tiny bit. So I do love them. They're near to dear in my heart. They're still up there on the internet, but it might be good to update some of them with a newer style. And yeah, because I think that the one thing that I want to do more of going forwards too, somebody just asked if I'd start a podcast where I interview scientists. I want to do more video interviews with scientists. I have so much fun going out and interviewing scientists. And so I really, really want to do more of that, where I go out and I talk to somebody who's had this recent big discovery or something come out, and I get to hear the science from them and help them sort of share their science with the world. So again, long sort of babble answer, but I would love to, I wish I had the confidence to reach out to those scientists who are doing the science that I was talking about more in the past, so that I could get more of that sort of scientist hands-on experience. And interviews. I love interviews. I love interviewing people. I just love talking about people, or talking with people about science. And so interviews with scientists are a great way for me to do that. Okay, in the last 3 minutes, I can't believe you guys have stuck around so long. You're all wonderful. Thank you so much for I was so worried that I was going to sign on and there were going to be 0 people watching now, and it was just going to be me babbling. Oh, okay. So here are the 2 last questions. So somebody just said, I have great teeth now, looking at my old bite-sized videos. I do, and I actually got a question from someone asking if I would talk about my Invisalign experience, because they had also done Invisalign. And I get this question a lot, and I've never like addressed it on the internet. I could just babble at you about how much I loved Invisalign for like 30 minutes. So my short answer is going to be that it was very expensive, and I saved up for a while. And again, as with I have no student loans. My parents were also helpful. I was still on their health insurance. They helped out with some of the like filling work that I needed to have done that went with it too. So it was expensive, and I had help with that, even though I'd saved up for a while. So that is the only con that I have about Invisalign. Everything else was amazing. My teeth were like caving in. I was breaking my teeth. It's this whole long big thing. But other than that, it has changed. My teeth don't hurt when I bite into food anymore. That's the number one thing. My teeth don't hurt anymore. So I went to an amazing orthodontist. It took like a year. I brushed my teeth like 400 times a day during that year, but it was 100% worth it. I can bite into an apple again. Amazing. So yes, I could make a whole video about how much I loved Invisalign, but I won't do that right now. And then finally, I just saw, oh yeah, so I think this is fitting. What would your post-PhD videos be centered, or would they be centered around genetics and biology, or are you open to exploring other fields too? I am totally open to exploring many other fields in other than genetics in science, but I personally love genetics, and I think that there are so many amazing genetics and biology stories to tell. And those are also the stories that I feel most comfortable and confident talking about, right? So I have the most expertise in these areas, and so I feel like I can do the best job of interpreting those stories better than I can for something in, say, physics, where, I took some physics classes a long time ago, but I'm not a physicist. And so when physics stories come along, I can do my best, but I might not understand as many of the nuances as someone who's trained in physics would. So I have so many amazing genetic stories that I want to go and film and tell after my PhD, but I just have to finish my PhD first. So I guess that's kind of an ending note, is that I'm gonna clean all this up, and then I'm gonna go to lab to keep working on my PhD. So I so appreciate that you guys have all stuck around, both for today in this one live stream, but also many of you have been around for years, and I'm so thankful that you guys have stuck around despite me like going off the wall and just like MIA sometimes. Like, I really appreciate, oh, my light battery ran out, or my light broke. Okay, all the things are telling me that I should stop this right now, but I will just express one more time, just thank you guys for sticking around here. Thank you for your time, and I so appreciate that you guys continue to support me and the crazy stuff that I do online. So that is it. Go forth and do science. And now you're probably going to watch 30 seconds of me figuring out how to turn off the live stream. So yeah, thank you for being awesome. Maybe I will do it again. Yeah, so if I do it again, I'm going to get a real webcam though. Okay. Now, yes, go forth and do science, and maybe this button will stop it. We will see.